Hello dear friends, and welcome to this video today in our YouTube channel. In this video we shall be looking at the correction of the Northwest 2018 paper 3 biology. So guys, stay tuned because we shall be highlighting all the corrections that could be made and ways in which we can remedy all the errors that will prevent students from scoring all the marks. So friends, don't forget to like the video, subscribe to the channel if you want so many contents. We have hundreds of contents already in our platform, guys. We are almost clocking 1000 contents, so make use of them, guys, and feel free to leave your comments in the comment section down below if anything you're not understanding, and we are going to reply to them and explain very well as soon as possible. So friends, share the video out with your friends so that they can have access to good contents like this, so that they are going to pass their own exams, friends. And alright, let's get started, guys. Let's get started. So this is the question we have here on screen. The question is on experiments. So it reads that macerate and form a solution of the muzzle, liver and gizzard of the bird used in question 3 above. So guys, we uploaded the video for question 3 already, which was talking about the dissection. Put in 3 separate test tubes, investigate the distribution of the enzyme catalase in each of tissues. Make a report of your experiment in the table below. Okay guys, from the question we see that this is to investigate the action of the enzyme catalase, right? Alright, so guys, let's have our table. Good. So this is a table of this form, guys. We have the procedure, observations, and we have the inferences. So guys, let's start. Under the procedure, we'll say that equal quantities of liver, gizzard, and mussels were macerated separately using a mortar and a pestle. So guys, keep in mind that this was supposed to be A, friends. So guys, keep in mind that the word macerate simply means to pound. Okay, the word macerate simply means to pound, but you are advised to use the word macerate and not to pound. Good. So we are going to equal quantities of the liver, gizzard, and mussels were macerated separately using a mortar and a pestle. Good. Then now guys, 6 mils of water were being added into each of the macerated organs to form a solution. So friends, I've used 6 mil. You can still use 4 mils or even 7 mils. It depends on you. Good. Then now the resulting solutions were filtered into separate beakers using a filter paper and a funnel. Good. Then friends, 2 mils of each solution was put into separate labeled clean test tubes. So friends, you notice that after macerating the liver, the gizzard, and the mussels, right, you'll be unable to make a solution out of it if you don't add a certain amount of water. But you should keep in mind that the amount of water that you add for the liver, for that's the liver, the gizzard, and the mussel has to be the same. Okay, it has to be the same. You must make it to be the same. Like for instance, you cannot be adding 6 mils of water in the liver and adding 4 mils of water in the mussel. That will not be proper, friends. So you add the same quantity of water. Good. So then now we are going to put them into different test tubes that have been well labeled. The reason for doing so is that we don't want to confuse which of them is which. That is why we must have to label them so that we don't get confused. Good. Then friends, 4 drops of hydrogen peroxide was added into each test tube. The test tubes were allowed to stand for 3 minutes on a test tube rack at room temperature. Good. So friends, after adding the hydrochloric acid, the hydrogen peroxide, you are going to experience some fizzing that is going to be going on, and we are going to allow it to stand for 3 minutes, after which we are going to observe and we draw our conclusions based on it. So guys, assuming that the 3 minutes has elapsed, what's our observation? What do we expect to see? We'll see fizzing. Fizzing occurred in the test tubes, in all the test tubes, friends, all of them. Then now the highest fizzing occurred in the test tube containing the liver. Good. And guys, the reason why the highest fizzing was seen in the liver, we could indicate it by simply just plus signs. The higher the number of plus signs here, the higher the fizzing. Okay, Fizzing, you can reason fizzing as the same thing like effervescence, okay, like effervescence, that's bubbles of gases. Good. So the highest fizzing occurred in the test tube containing the liver, that is this, and the reason for that is that the liver is the highest metabolic active cell in the body of the bird, okay, even in our body as well. So it has to contain the highest amount of catalase enzyme. Good. Then friends, the higher fizzing, that is the second, was seen in the muzzle. Great, it was seen in the muzzle, guys. Then now the high rate as was seen in the solution containing the gizzard. So the highest was the liver, the higher was the muzzle, then the third was what, the gizzard. Good. So friends, these are your observations. Then let's explain the meaning of all what we just wrote. That's under our inferences. So we'll see that active tissues produce hydrogen peroxide as a byproduct of tissue respiration, but this hydrogen peroxide, guys, cannot be allowed in the body. So we see hydrogen peroxide is very toxic and must be converted into non-toxic products. Good. So we are now going to use the catalase. Now is an enzyme produced by all living cells that help to break down hydrogen peroxide into water and oxygen. Good. So friends, since due to the toxic effect of this hydrogen peroxide, now the catalase enzyme now, which is produced by all living cells, now helps to break down this toxic hydrogen peroxide now into forms that are not toxic, such as water and oxygen. So let's have the equation. We have hydrogen peroxide being broken down, we have into oxygen, and we have water. Good. So these are the, I don't think the question is balanced first. I don't think it's balanced. Let's see. Okay, we just need to add a 2 here, friends. If we can add a 2 in front of this hydrogen peroxide and also add a 2 here, I believe everything will be good, right? Just add a 2 in front of this hydrogen peroxide and add a 2 in front of water. Good. That will solve it. Then friends, fizzing indicates the release of the oxygen gas. Great. Then now all the above organs produce hydrogen peroxide as a byproduct of metabolism. Good. So now guys, we should know that once we must have added the hydrogen into those solutions, we are going to experience fizzing, like I indicated initially. We are going to experience fizzing coming out of it, and the fizzing is as a result of the breakdown of this hydrogen peroxide to give us what, oxygen and water, because like that hydrogen peroxide is toxic, right, and it must be broken down into forms that are not toxic by the catalase enzyme. Good. So now we say the highest fizzing occurred in the liver because it is metabolically very active and it is the site for most metabolic activities. Good. Then now friends, the gizzard is also very active in metabolism, in mechanical digestion, while the muzzle helps in movement. Great. So friends, this is all about the experiment. This is all about the procedure, observation, and inference, guys. This just what we have written so far can give us about 15 marks so far, guys. Can give us about 15 marks or more. Great. So the key point you should take note here is that this procedure, okay, the procedure simply involves all what you did that led to you transferring the different solutions into their test tubes and labeling them. Okay, that's what should come under the procedure. Then now your observation, what you saw at the end of the time you gave here, what you saw at the end, like our time was 3 minutes. What did we notice after 3 minutes? This is what we need to write. Then our inference, now we are simply trying to explain the reason for this observation, the reason for our observations. So friends, don't forget to like the video, subscribe to our channel if you want more content, guys. Thank you very much. Let's look at the next part of these same questions. Let's look at it. So the next part is saying that what conclusions can you draw about the amount of catalase present in each tissue. Good. So friends, let's not waste time. We'll simply say that the quantity of fizzing in each tissue tells us the quantity of catalase enzyme that is present, okay? Because we said initially that that the liver produces what the highest effervescence or the highest fizzing, right? Therefore it contains the highest amount of catalase. So friends, in accordance to this point now, it's simply repeating what we said initially, right? Good. Then I will say that the rate of metabolism is highest in the liver and thus has the highest amount of fizzing. As a consequence, guys, it has the highest amount of what? Catalase. Good. Highest catalase. So you could remove this fizzing and you simply put catalase enzyme. You're perfectly correct. Good. Similarly, now the muscles have the higher metabolism and thus has the higher rate of fizzing or the higher rate of catalase enzyme. Still works. Then now we start the gizzards have a high rate of metabolism and thus produce a high rate of fizzing. Good. So friends, that's a direct point. In the lab, friends, as you're doing these things, you'll be seeing how your answers will be coming out just immediately. Okay. Then now we'll say that all the tissues contained catalase and the thus all produce the hydrogen dioxide as a byproduct of metabolism. Great. So friends, I think all these points are direct, right? All these points are direct points. The points are simply explaining what we did initially. The points simply want you to give more meaning to the experiment you just did. So in the laboratory, you're going to see all the observations we listed initially, and you're going to see, provided you make the experiment so well. Great. So friends, let's look at the next part now, the last part, which talks about the precautions. Let's look at it. Okay. Next part is asking that what precautionary measures can you take to ensure accurate results? Good. So friends, let's not waste time. We'll simply tell them that each of the syringes and test tubes were thoroughly cleaned using the test tube racks and rinsed with the test solution. So friends, likewise, like what we know in chemistry, that when trying to fill the burette with a solution you have to titrate something with, you have to rinse the burette with that solution. It's the same thing here, friends. If you want to transfer the solution from the beaker into the test tube, we must have to rinsing the syringe we have to use using the solution in which we want to transfer. Good. So we have to rinse it such that we are so sure that no extra amount of water has been added to it. Okay, has been added to the solution. Remember, guys, don't forget that we are to add equal volumes of water. The moment you don't add equal volumes of water, your results will become faulty. You won't have good conclusions to draw at the end. Good. Then friends, the next point will simply tell them that the same quantity of the different tissues were used. So friends, like I said, in one test tube, if we are choosing to put 4 mils, right, in each of the test tube, we must put 4 mils. We are not going to put 4 mils in one test tube, then the other test tube we put 3 or even 3.5. No, guys, we put the same amount to ensure accuracy. Good. Then the next point, guys, will simply tell them that the test tubes were made to be of the same size and height. Good. So friends, if the test tubes are made to be of the same size and height, we'll be able to analyze what exactly has happened. Okay, we're able to analyze the level of their fizzing. But if one test tube is thin while the other is fat, when both of them have the same size, guys, it will be so difficult for us to determine what exactly has happened. Okay, so to conquer this shortcoming, all we simply do is to make sure that all the test tubes are of the same size and length. Good. Then friends, the next point will simply tell them that equal volumes of the tissues were used. Friends, like I said, this point is super important, very important, because if, for instance, you use 4 mils of the solution, I use 2 mils of the solution, our results at the end of the day can never be the same. Okay, they can never be the same. They will be faulty, rather. They will rather be faulty. So in this case, guys, keep in mind that we are dealing with enzymes and substrate, right? Enzymes on the substrate. So we must have to be very, very careful with the amount of each product that we use. So for instance, the last point, we'll simply tell them that equal amounts of hydrogen peroxide were added into each of the solutions. Very important, friends, very important. So these are the points that are going that you have to follow in order to have high accuracy. Okay, these are all the points. So friends, don't forget to like this video, subscribe to the channel, give it a thumbs up. That is going to help me to keep producing more and more contents for you. Thank you very much, guys, and good luck to all those who are writing. Stay safe, and God bless you.